So everything that you see here in terms of proteins, they will have in some sort of an enzymatic activity. So one of the proteins that we will look at this semester is called chymotrypsin. And that we're not going to talk about until after the first test. Okay, so chymotrypsin, trypsin, those are all proteins that break those peptide bonds. Okay, so they're going to break those peptide bonds. So they belong to a class called serine protease. And they classified not just chymotrypsin, they have found that the active site that binds to these particular enzymes have serine in their active site. Okay, so you have a protein or an enzyme, and then that enzyme binds to a substrate. Okay, so when it binds to a substrate, then you have some sort of a reaction happening. In this particular case, you have some product being released. So that's kind of the simple version of what an enzyme does. So these classifications, the serine protease enzymes have actually 3 amino acids within the active site. Right? So you have an enzyme, it's going to bind to a substrate in its pocket, right? So it's like a lock and key. It binds, and then a reaction happens, and you get product released. So this semester, we're actually going to take a peptide sequence, have you throw in a particular enzyme, and you tell me where it cuts, because it's very specific where it cuts. And then you come up with the puzzle as to figure out what the sequence is, short sequences. Next semester, we'll actually be looking at how it happens. Okay? So this is the how question, how does it happen? And so with the active site of these specific serine proteases, like I said, there's 3 amino acids, one of them is serine. Right, so how do I know it's serine? There's my OH group, right, the functional group. So then we also have histidine, and this is going to be our aspartic acid. All right, so there's actually, oh, there's my aspartic acid. So, you have these 3 amino acids, which is very important within the active site for catalysis to occur. So if you mutate one of these amino acids into something else, because histidine is positively charged, and you just change it to say aspartic acid or glutamic acid, it's acidic, it's going to change the properties, right? And you can see that. You can see the reaction right now. I'm not going to go through the reaction, but obviously if you change that key amino acid players, those 3, you're going to see that this particular enzyme can't be active. Does that make sense? Okay. So we're going to look at, so obviously this is important because if you know the primary structure, you can figure out, after it folds, you can actually figure out what's on the active site. Another thing we're going to look at is also disease states, like hemoglobin, for instance. And hemoglobin has many different types of mutations that occur. And one of the things that happens with sickle cell anemia, and we'll dive into more detail on this, but I want you to get a glimpse of how important these amino acids are. We have normal hemoglobin, which is glutamic acid as this group. At the beta chain, there's a glutamic acid. In people with sickle cell anemia, there is a mutation in the gene. And that gene gets converted. Once it gets translated, it becomes a valent. Glutamic acid, polar or nonpolar? Got a 50-50 chance. Polar or nonpolar? Polar? Valine is nonpolar. So changing this one amino acid from a polar to a nonpolar completely changes the structure of that hemoglobin. And that's going to cause what's called sickle cell anemia. You really need to know those amino acids. They just have to come very quickly. Okay? 1-letter code, 3-letter codes, and polar, nonpolar structure-wise. So the next 2 slides is just pretty much what I've just said. I'm giving you examples from the previous slide. So protein structure, different proteins can be compared to get knowledge, like with the serine proteases. If you know a structure of an active site, then you can actually classify different ones into into the same group. It provides a lot of information about the evolution between bacteria and eukaryotes, for instance. There is similarity, and you'll see that when we get to replication, transcription, translation, we learn a lot from the bacterial system because it's simple, it grows very quickly, and we can get a lot of information from these small organisms, and they were able to figure out that the process was the same from eukaryotes to eukaryote system. It's just there are more enzymes involved in the eukaryote system. You can actually figure out some internal repeats, and that goes back to specific sequences. It doesn't necessarily have to be within an active site region. It could be other parts of that protein region that's important. And you'll see some of that sort of examples with histidine, because histidine is found in hemoglobin, and that's going to be an important amino acid. And that, through evolution, remains the same. That repeated sequence remains the same. Okay, so protein structure function is very important, like I said, and that goes to trying to figure out certain diseases, right? Just based on changes that occur within the primary structure, you get a certain disease. So it's not just sickle cell anemia. I'll show you in table when we get to that stage, but there's lots of different diseases just by changing these amino acids. Not just polar to non-polar, but say you have a small amino acid, it can get bulky, put a bulky amino acid, just